The endoplasmic reticulum, or ER, and the Golgi apparatus produce and process proteins. These proteins move from their site of production in the ER through the Golgi, after which they proceed to their destinations, either taking residence in the plasma membrane, or being secreted outside of the cell, or fusing with lysosomes. Proteins from the ER travel in transport vesicles to a region of the Golgi called the cis-Golgi network, where protein modification begins. We can follow the maturation of this compartment by marking it with a dark color. Vesicles fuse with the cis-Golgi network until it begins to mature and move into the Golgi stack, and then vesicles create a new compartment in its place. The compartments, with their proteins, mature to become the medial and then the trans compartments of the Golgi stack, where further protein modifications take place. The compartments of the Golgi gradually mature and progressively move through the Golgi in the cis to trans direction, carrying their contents with them. The trans-Golgi network acts as a sorting and distribution center, directing molecules to their final destinations. The proteins leave in vesicles, which bud off until this final Golgi compartment disappears and is replaced by a maturing compartment immediately behind it in the stack. The transport vesicles that are associated with the Golgi apparatus function to return Golgi resident proteins back to earlier Golgi compartments for reuse. The mechanism by which proteins move through the Golgi apparatus has been a long-standing area of controversy, but the model described here has considerable support from recent studies.